and welcome to another Pype pod. Today we're going to be continuing the anatomy physiology theme that we talked about, and this time it's a two-part series looking at the lymphatic system and immunity. I didn't want to give you guys an hour plus anatomy physiology because I know that is way too much to absorb in one time, so I've broken this one up. So without further ado, let's kick straight into it, and we'll start by looking at the lymphatic system itself. We always talk about lymphatic system and immunity in the past. A lot of the focus goes on to the immunity aspect, and rightly so. It's far more interesting and a lot more complicated, so a lot more points to sort of get your head around. But it's the lymphatic system that actually is the housing for your immune system. Okay, one of the main roles is that it's the protection of the body that it has by allowing certain substances to go in it and moving waste products from the interstitial fluid back into the circulatory system after it's been cleaned. It also has a role within the transport of your dietary lipids. And now dietary lipids are used throughout the body. Okay, all sorts of fatty acids and lipids are used throughout the body in forming cellular structure. So they actually play a very big role in the production of hormones, the production of cells, and a lot of the time the stabilization of your body. So it doesn't really get enough credit, the old lipids. They're usually associated with negative connotations of getting fat, but they are absolutely vital in the function, and it's the lymphatic system that transports them around to different parts of the body. Obviously the main focus of the lesson will be focusing on the third, which is the protection, how the immune system and the lymphatic system work together. The lymphatic system is made up of a whole bunch of vessels. Like the vascular system, it has the same three layers within each one of its vessels. Okay, and now these, like blood, are pretty much throughout the body. Okay, so they complement capillaries, they run alongside all your vessels, and they're right through your body in order to gather fluid from the interstitial space. Now there's a few areas listed that you can see that doesn't involve lymphatic tissue. Okay, so anywhere that there isn't any vascular structure, obviously won't have it. The central nervous system itself doesn't need it as well. It's a pretty well protected system. And then you've got areas of the body that are more focused in the production and storage of blood vessels, like the splenic pulp and the bone marrow, that also don't require the lymphatic vessels around them. We'll talk more about the bone marrow within the next lesson itself. Now, the general flow of lymphatic tissue is the capillaries pick up anything that leaks out or is too much, if you will, within the interstitial space. So if you remember way back in the original lessons of fluid physiology, you'll know that based on hydrostatic pressure, fluids will move outside of the vascular system into the interstitial space. Now, as that area builds up, becomes edematous or even slightly edematous, the lymphatic capillaries work on the same principles as osmosis, diffusion as well. So they have their own internal hydrostatic pressure, and they work either on a concentration gradient or an Ohm's drift gradient as well to draw fluids in. From there, no surprise, capillaries become vessels, but then we have little stop points in between the vessels called lymph nodes. Okay, and we'll talk more about them soon because they're a massive role in our immunity. Eventually we get to what's known as the lymphatic trunk, and then collecting duct into the circulatory system again. Now we'll cover a little bit more of that with diagrams to help explain how that whole process works. Okay, so when we look at the drainage aspect, obviously all that excess fluid that's found in the lymphatic system goes back into the circulatory system via the subclavian veins. Right next to the junction of the subclavian and the jugular vein is a portal or a duct that you'll find within the lymphatic system. Now the thoracic duct is the one that's responsible for draining, for lack of a better term, the main portion of the body. Okay, and the right lymphatic duct does the right upper torso. What happens is as this fluid builds up to a high enough pressure within the lymphatic system, it travels back up towards the subclavian arteries and the jugular vein junction, if you will, that you see there in the neck. And then it has what's known as a one-way valve. As the pressure builds up, the one-way valves open and the excess fluid is dumped back into the central circulation. Okay, now there are areas within the vessels or around the capillaries that have similar leakage back into them, but the majority of the lymphatic vessels overflow is moved back into central circulation. That way, if it's too much for the entire body, it can go through its normal process of being filtered through the kidneys and reducing the overall circulatory volume in waste products. Okay, so that's what happens. And it only happens with pressure. So the lymphatic vessels need to maintain their own base hydrostatic pressure in order to function, in order to stay open, and in order to encourage more fluid and waste products in order to draw into it. Okay, so that's basically how it works. Now, a point that I didn't mention, and I'm sure you're all aware of it, is the lymphatic system doesn't have a pump like the circulatory system. Okay, so it doesn't circulate around the body. It is influenced by muscular movements. It is just evenly distributed throughout the body. And as it builds up, dumps into the circuitry, the central circulation, and then again, it's evenly distributed. More activity you have, massage, muscle activity, skeletal movement, that obviously encourage movement of the lymphatic system, but generally speaking, it's just a somewhat of a static system. When we look further down into the lymphatic system, we understand that it needs to be supported by certain organs. Now, I use organs in the very loose term, not talking about larger anatomy organs, but organs in the sense of areas that support the lymphatic system. Now, red bone marrow and thalamus we'll talk about in the next lesson more. We've already mentioned lymphatic nodes, and we'll cover a little bit more off on those. Then we have these little things called lymphoid nodules as well. So they're not true nodes, but they still have a similar function. Now, the spleen as well, in the lymphatic system sense, your largest lymph node that we have. Okay, so it's your main filtration organ of the lymphatic system. And a lot of the time it's where immune cells will actually be housed in order to conduct their function without giving too much away too early. Now, focusing on your lymph nodes, this obviously plays a big role in clinical presentation. They're found in clusters throughout the body. So we have areas like your ankle, your groin, your armpits, your neck. Okay, there's large clusters of lymphs. Now, the reason that is because like the circulatory system, these areas are involved in crossroads of where lymphatic vessels come to. Now, what these do is they're a filtration plant, so they purify your lymph. Okay, the fluid within the lymphatic system is called lymph, and the lymph nodes are responsible for purifying that. The way they do it is taking advantage of their structure. So as we can see here in the picture, we have a lot of vessels coming in, so in this case 4 vessels coming in to the lymph node, and then 2 vessels coming out of the lymph node. Now, within the actual medulla of the lymph node is where immune cells will collect and congregate. Okay, so in this case we actually have activated monocytes. So macrophages, as they're already activated, will be found in this area in order to aid in that phagocytosis that we'll talk about. We also have specific lymphocytes, like T cells and B cells, that you'll find in the lymph node. Now, the advantage that it plays of having 4 in and 2 out is it actually increases the amount of time that the lymph is in the lymph node. If it had equal in and equal out, then there's a chance that your lymph wouldn't be cleaned by these immune cells, wouldn't go through phagocytosis to remove all the non-self, all the particles, debris, that sort of thing. So it takes advantage of drainage in order to optimize time for these specialized cells to capture things. Knowing that, it's kind of easier to figure out why during times of infection, when we have an increase of waste products, pathogens, that we end up with a symptom of lymphadenopathy. So it basically goes into hyperdrive and becomes engorged with lymph fluid and immunity cells. And we can palpate them as a clinical indicator of potentially an infection or an increase in waste products. Now, apart from those classic lymph nodes that we find, we'll also have those lymph nodules, as I alluded to. Now, what they are is they're more specialized single groups that basically form the same function, but they're not nodes. Okay, they hang out within the mucosal membrane of vital organs, your respiratory tract, urinary, digestive tract. You'll find these lymph nodules. Okay, and then the way they differ is they're given the term MALT. So they're associated with mucosal membranes, not lymph circulation. Now, the classic ones that you would have heard of before is the pilus patches. They're around the small intestine, and your tonsils. Tonsils aren't lymph nodes, they're nodules. And we also have it in the appendix, which does prevent us from getting appendicitis every time something gets caught in it. Okay, so it's a very, very functional lymph nodule to have, but they have the same function as lymph nodes. They're just found in different sorts of tissue. I think, as an overview, that covers the lymphatic system. Like I said, it's relatively simple system structurally to get your head around, and functionally the sort of things that it does. But the main role and the main complexity comes when we start talking about immunity itself. Now, as mentioned, it's the lymphatic system that allows for immunity to occur, if you will, or allows for immunity to function better within the body, and be the focus of the rest of this lesson and the next one itself. So when we talk about immunity, what we're basically talking about is a resistance to a disease, resistance to pathogens. Now, the immune system is broken up into two different sorts of defense systems. Okay, so we have an innate and an adaptive, also known as a non-specific and a specific. Okay, within the innate system, it's broken up into your first line and second line of defense, and when we get to adaptive, we move into our third line of defense. Now, when you start studying the immune system, it's very easy mistake to make that we start thinking about innate and adaptive as two different systems, or start thinking about T cells and B cells as two completely different systems. But what I want everyone to keep in mind right through this whole presentation and the next one is that these things work together. And if it wasn't for the innate immune system and certain functions within it, the adaptive immune system wouldn't even function. Okay, it wouldn't even be activated. So they are deeply entwined. So remember that, please, and always draw back to it and ask yourself, how did this line of system actually activate? Or how did the previous one help in the activation of the next one? So looking at natural resistance to a disease. Okay, it just means that you have the ability to either not catch the disease or easily fend it off. Okay, that's what the term resistance means. Now, if you have a non-specific resistance, that system is responsible for a huge variety of pathogens. Okay, as opposed to a specific one which focuses on an individual pathogen. Now, when I use the term pathogen, what I mean is it's some form of microorganism that has the ability to cause a disease. Okay, that's what the term pathogen means. And obviously, the immune system works in resisting attacks from certain pathogens. Now, every species is immune to certain pathogens of another species. Okay, so microorganisms can affect different species and have no effect on humans. So naturally, based on our genetic code, we are naturally resistant to certain pathogens. Okay, we're certainly especially resistant to a lot of microorganisms. Okay, remembering that we have a massive homeostatic relationship with microorganisms, to the point that their genetic material outnumbers ours within our body. Okay, in every cell that is you, there's around 100 plus cells that are microorganisms. So there's obviously an inbuilt natural resistance that we have to microorganisms. Otherwise, our body would be constantly inflamed. and attacked from our natural flora and what is automatically found within our bodies as well. First line of defense is your innate defenses. Now this is just biological structures within our body that is designed to protect the infiltration of pathogens. Now there's quite a few within our body, and you can really get deep into the first line of defense, but just as a summary to go over it, the main first line of defense that we have is our skin. So that provides a physical barrier of dead cells, okay, and it's very difficult for microorganisms to penetrate through these cells. Now, as an added bonus, we have things like sebaceous glands that produce sebum, and that itself is quite acidic and kills a lot of microorganisms found on our skin. Okay, so our skin is waterproof, thick fibrous protective layers made by keratin, so it's very difficult for pathogens to take advantage or infiltrate our skin itself. Now if for some reason it does, it bypasses our skin, so it goes through one of our natural portals that are already open, then within our bodies we have natural immunity as well. So a lot of our respiratory tracts and other entries have a mucosal layer. Now what a mucosal layer can do is hold pathogens at bay for simple second line defense to take care of it, or just the natural environment of certain mucosal layers to take care of it itself, like the vagina being naturally acidic to kill microorganisms. Okay. Now on top of that, we have nasal hairs, okay, which acts as a filter for a lot of the larger particles that contain pathogens. And then we have smaller hair-like structures called cilia. Now these are found all along our respiratory tract, the main bronchi, and it's also found in our ear. And they're involved in moving, wave-like movements, in pushing pathogens back up so we can cough or sneeze them out, or pushing them back up to the top so they can be ingested within the stomach to be broken down by pepsin and hydrochloric acid. All these things work together. Now, naturally, within our mouth and our tears, we have a enzyme called lysosomes, which we'll learn a lot about as we go along, and they're also designed in the destruction or the breakdown of microorganisms. So all these things are just naturally in our body, naturally in that first line of defense, protecting us from microorganisms or potential pathogens. We don't even need to activate the second or third line most of the time, because our body and its survival adaptive ability that we have naturally takes care of a lot of it, and that is what's known as your first line defense, or the first level of your innate defense. Now if for some reason your pathogen breaks through, or there's too much numbers, somehow it has gotten into the body and it started to reproduce, it has started to break down your body, use your body as its source of medium, we have a second line of defense that is also non-specific. Okay, and the things listed in the second line of defense are your inflammatory response, your phagocytes, natural killer cells, antimicrobial proteins, and a fever. Okay, so actually having a fever as well. So what we'll do is we'll go through each one of these second line of defense and we'll explain what happens in each of them. Because if we don't understand the second line of defense, then we won't actually be able to activate the third line, and therefore understand the complexity of what happens when we get to specific immunity. So firstly, looking at the inflammatory response. Now I understand this slide is busy, but trust me, a lot of you guys know this already. Now, when a cell is injured, it doesn't have to be a cut. I know we talked about a cut last time, but whenever cellular tissue is damaged, internally, externally, doesn't really matter, processes start in order to protect itself, or in order to optimize other processes in order to clean and then repair as well. So the first thing that happens is once the tissue is damaged, that particular cell or tissue releases a whole bunch of chemical mediators. Okay, now these chemical mediators, they attract leukocytes and they cause vasodilation. Okay, so they cause this vasodilation effect. Now, the main chemicals at play is your prostaglandin. Okay, so prostaglandin is the main chemical mediator released, but other ones include your kinase and your histamine. Now, each one of these work in dilating the actual area, therefore bringing more blood to the place. Now, the only exception to this is in the instance that the vessel itself is cut, so therefore you're physically bleeding. Now, we talked about that last time, and that will release very specific and very centralized vasoconstriction to that area, but then vasodilation to the surrounding area in order to bring more things in whilst holding the vessel whole as small as it can be. So it gets a little bit complicated with that. So I just wanted to mention that because I know the last lection I talked about a similar process that causes vasoconstriction. Now I'm saying vasodilation, so I just wanted to make that distinction to let you know that both occur in that process. As a result of this vasodilation, a whole bunch of things happen. So within the vessels itself, we get more blood to the area. So the area itself becomes hyperemic. So lots and lots of blood to that area. And that's where our heat and our redness comes from. Okay. Now this brings a lot of repair properties, a lot of oxygen and nutrients to the area, so it can actually go through and repair itself without building waste products. We also get the capillaries leaking all the plasma out. Now, the advantage of leaking all the plasma out as well is any form of phagocytosis or cellular debris that has happened as a result gets leaked out and carried along with it for the lymphatic system and the lymph nodes to take care of it later on. Okay, so all this good stuff happens, but as a result of the leakiness, it causes temporary edema. Now, with the edema, we get the pain and the swelling. Okay, so obviously our classic signs of inflammatory response is heat, redness, pain, and swelling. As we get the vasodilation and we get the increase of tissue and the cleaning out of the solution, what else these chemical mediators do, and sometimes more importantly within the entire immunity response, is it actually attracts leukocytes. So it attracts your white blood cells to the area in order to help clean up and start the process in an active immunity. Now, I won't give too much away in this particular one, but just so you know that when the inflammatory response happens is actually the first signal for leukocytes to encourage that phagocytosis and in turn activate your adaptive immunity. So looking at phagocytosis itself. Now, phagocytosis is a term that means phagocytes or leukocytes coming in and engulfing a pathogen or engulfing a substance. That's what phagocytosis means. Now, it is broken down into 3 phases. Now, those 3 phases start, number 1, as I already talked about. Those inflammatory mediators create what's known as a chemotaxis. Now, chemotaxis is a signal. It's a signal that gets to the bloodstream in order for the phagocytes to come along, move through the vascular or lymphatic system into the tissue drawn by these chemotracts or these inflammatory mediators. Once they get to the area of concern, the area of tissue damage or infection, they go through adherence. Now, adherence means that they find the debris or they find the pathogen and they go through a process called opsonization. Opsonization is when that debris or that pathogen links to the side wall of the phagocyte itself. Once it does that, then it goes through ingestion. So it holds on to the pathogen, it extends its cellular membrane around the pathogen, bringing it inside itself. From there, it has neatly packaged capsules of lysosomes that it pushes into the capsule holding the pathogen. Now, lysosomes, as we mentioned, the enzyme, there's a few other enzymes in there, but the enzyme that breaks down the pathogen, making it a bunch of cellular structure. From there, it excretes it. Okay, it dumps out this cellular waste. Now, the cellular waste is picked up by the lymphatic system goes through the process, cleaned a little bit more within the lymph glands itself, back into the circulatory system, and if it's not used, if the cellular structures aren't used within the body, it gets excreted through the kidneys. Okay, but a lot of this cellular waste is recycled, okay, picked up by the liver, utilized in cellular metabolism, so we use it as an extra bit of nutrients as well. Okay, so the whole process, remember, everything works together in hemostasis, and that's phagocytosis for you. Remember, it is drawn in by your chemical mediators, making that chemotaxis. Now, the first leukocytes on the scene is your neutrophils. Okay, so your neutrophils are the first ones to exit the bloodstream, or the lymphatic tissue, the lymph itself, into the tissue, and then it goes from there. The cell that follows from there is your monocytes, because remember, monocytes have to be activated or mutate into macrophages before they're useful. A monocyte cannot go through phagocytosis. A macrophage is one of the greatest cells at phagocytosis, but that process can take around 12 hours. After that, with the macrophages, then we start getting our specific cellular structures coming into the environment, sometimes, which is your lymphocytes. But you'll cover more of that. I just wanted to give a teaser to let you know the process as it goes in. Another cell that we have that helps out with phagocytosis is what's known as a natural killer cell. Now, the reason they are defined as natural killer cells is they are still non-specific killers, but they're a type of lymphocyte. So lymphocyte, if you remember, is responsible for making T and B cells, but some of its development will produce natural killer cells. Now, the advantage that natural killer cells have is they will just freely roam the body, and they will kill anything that is identified as a non-self. And then themselves can produce chemicals to enhance the inflammatory response, to make the inflammatory response more prominent, to bring more things to the area. So if we imagine, this is how it's different, is if we have a tumor cell, or we have a microbe within an actual cell itself. A phagocyte is not going to come along and engulf one of your body cells. It's just not what it does. Okay, but a natural killer cell is going to see that it's different, and then go through a process of lysis. It will land on the outside, and it will cause that cellular structure to break. Now, with that breaking of that cellular structure, that's a cell that's damaged. So that will start an inflammatory response to help come along and clean up all the debris. Okay, so a natural killer cell is an assistant to your body that will go around killing non-specifically anything that it doesn't recognize as self, and then start the inflammatory process, which in turn can start the entire immune response. So that's what natural killer cells do. Okay, so as far as lymphocytes go, I think they're equally as cool as the other two. Now, other things besides leukocytes, okay, so we have other things in our bodies that help us in our second line of defense and removal of microorganisms. So it doesn't always have to be immunoglobulins and your leukocytes that work with your immunity. We have other specialized substances that work at destroying microbes. So one of those that you may have already heard of before is interferon. Now, if anyone studied any form of pharmacology, you'd realize that interferon is a drug that is given for viral infections. So same too as this, it's also an antiviral protein is what interferon is. Now, the way it is activated within the body is pretty cool. So what happens is a cell that is infected with a virus will release interferon from itself. So as a form of distress, it will release interferon. Now, when the neighboring cells are activated by interferon, they actually produce antiviral proteins. Now, these antiviral proteins enhance that opsonization. So they enhance the ability for phagocytosis to happen. So they make it more obvious that this cell here with the virus in it needs to go through a process of phagocytosis. Okay, so that's what interferon does within the body. Another type of antimicrobial substance is what's known as a complement system. Now, it's exactly what it sounds like. This complements the immune response. Now, throughout our body, we have about 10 to 20 proteins that just float around our body and they're not activated. Okay? They only get activated when they touch the cellular wall of a foreign cell. When they touch the cell, they activate from there. Now, things that make it more likely to activate is your certain immunoglobulins. And we'll learn about immunoglobulins in the next lesson. But when they touch the cell, they can in fact draw this complement system to that cell. Okay, on top of that, the proteins themselves can help the allergic or the inflammatory reaction. So they can help the whole process by enhancing opsonization, inflammation within the whole process of immunity. So what they do is this protein comes along and will literally bust a hole through the phospholipid layer of the microbe itself. So it's mostly effective on your bacteria, but there are certain ones that can help out with nucleic activity or protein like viruses. Okay, but mostly it's bacteria. Once this complement protein pushes through the cell, it actually draws in more complement proteins. And what they do is they create a pore together. So all of them bust a hole in it, and all of a sudden there's a hole within the actual cellular wall of the microbe. Now, it's not just one. A particular bacteria can have hundreds of complement protein pores within it. As a result of that, the destruction of the integrity of the wall and the changing of hydrostatic pressures, normal osmosis, is a whole bunch of water will be drawn into the cell, and the cellular wall structure isn't going to be strong enough to hold it. So it's gram-positive bacteria. Just by creating pores is enough for the actual cell to fall apart. But if it's a negative, has less hydrostatic pressure, then it will cause fluid to flow in, and the same result will happen. We'll get lysis of that cell. So pretty cool, and it will play a role later on in the adaptive immunity as well. So stay tuned for that one. I know you're all very excited that that's going to happen. Okay, the last thing that we're going to discuss in the innate immunity is the role a fever plays. Now, a fever, so an increase in temperature within the body, is activated by pyrogens. Okay, now these are also known as pyrites. Pyrogens or pyrites are released by macrophages. So as the macrophages come in contact with the foreign substance, they release pyrogen. Okay, now if we remember back to the inflammatory and the chemotaxis of phagocytosis, 12 hours later, macrophages are formed. Okay, so that's why every time we get an infection, we don't end up with a fever. Okay, the other immune systems are able to take care of it without the need of the fever. But if the infection goes on long enough, macrophages to get involved, then pyrogens will be released. And a pyrogen will reset your hypothalamus. So instead of saying, all right, hypothalamus, I want you to sit around 37 degrees, it will say, I want you to reset to 37.5. So therefore the body will work together in order to raise the temperature and maintain the temperature at that higher level. So it's a new step of homeostasis. Now obviously, if pyrogens are kept being released, then we can start getting dangerously high fevers. There's no off switch for this, unfortunately. As soon as the pyrogen is released by these cells, it's going to go to the hypothalamus, and the chemical reaction is going to set the standard higher every time. And that's how it works. So when we have a low-grade fever, it obviously can be extremely beneficial because it speeds up our metabolic rate. Therefore, it speeds up the repair, it speeds up the cleanup, it speeds everything up, which means that we have the infection less time, less damage on the body. But it also causes the liver and the spleen to hold on to iron and zinc. Now, iron and zinc are used by microorganisms in their reproductive process. So essentially means that they're holding vital nutrients away from the microorganisms themselves. Now obviously, if the temperature gets too high, then it starts to have an effect on enzymes and proteins. Because as things are speeding up with the metabolic rate, there's an optimal range of effectiveness. If it goes too high, the speed degradates the enzyme and the proteins, and then they can't function. And that's when we start getting dangerously high fevers. That's when we start getting the effects of hypothermia and things like that. Okay, I think that will do for this particular lesson. So as I mentioned, this covers off the innate immunity and the structures of the lymphatic system itself. The next lesson, we're going to look at the adaptive immunity. So we're going to draw on things that we've learnt from this one and see how it complements in that adaptive process. And hopefully by the end, you have an overall full picture of the lymphatic system and the immune response. Until next time, take care.